Hello everyone. Today on the subject of medical microbiology, we are going to study about biohazardous waste. So it is a very crucial thing and you must understand that in research that's very important subject. So when dealing with the introduction of biohazardous waste, you must understand the meaning of the correct meaning of the word hazardous is poisonous. Okay, so when you are dealing with such poisonous materials, and you must understand from where they are generated, how they are generated, and how to deal with them. And the outcome of this topic will be that at the end of the story, you will be able to fully understand what are the techniques employed in the disposal of such uh, hazardous waste. So, what is biohazardous waste? Biologically derived waste or waste type contaminated with biological material is, is the definition of biohazardous waste and includes any biological process, whether medical, clinical, veterinary, or resulting from animal processing to produce biological tissue, fluids, potentially infectious disease. So anywhere, any place where such procedures are going on that are dealing with the infectious materials or the infectious conditions or dealing with the body fluids that are diseased and also the clinicals and hospitals and the laboratories combined where the treatment is going on and the research is going on. Any items or implements used in these processes have a high contamination risk. If left unmanaged, they can often become more hazardous with serious environmental and health impacts. So, such poisonous and such hazardous materials should never be left out in the open because initially and eventually, when it comes to the contact of living organisms, when it comes to the contact of environmental implications and all those things, they are going to affect everyone. Okay, per hospital bed per day in high income countries generate an average up to zero point 0.5 kg of hazardous waste. So what they're trying to explain to you that in a developed country, the if you take the example of patient on a hospital bed, so per bed they produce the waste material that is equivalent to zero point 0.5 kg, and conversely, when it comes to the countries which are developing low income countries, they generate zero point 0.2 kgs of biohazardous waste. So you must understand the scenario in which these waste materials are generated and how uh, are the conditions that are required to deal with such scenario. So where are biohazardous waste generated from? So where is the main source of this biohazardous waste and from where these things are generated? You must understand it. Okay. The type of organization generating waste include dental practices, general practices, health clinics, hospitals, opticians, veterinary practices, pharmacies, pediatrics, practices offering alternative treatments, private and independent healthcare organizations, research facilities, and all those things. So here you can very easily understand that all the things that are being generated as biohazardous waste material, they are somewhat directly or indirectly linked with the health practices system, health clinics, hospitals, and research laboratories. So, meanwhile, you can say that everywhere and everything that you observe and as a source of biohazardous waste generation source, they are all interlinked, they are all connected together, and they all have these probabilities of generating such poisonous waste material, and it should be dealt in a very particular manner according to its uh, hierarchy. What are some biohazards? Example. Biohazardous waste include human body fluids. Okay, see uh, what they have tried to explain it to you. Like they all have human body fluids. Okay, human blood and blood products, microbiological waste. Okay. See, whenever you are dealing with pathological conditions or any other thing like that, what you see and what you observe, the basic thing is that the basic scenario is that you are always dealing with the poison substances, you are always dealing with the disease conditions, and you are always dealing with potential infections. So everywhere the scenario is the same. Likewise, you have to deal with the specimen in a very uh, perfect order, in a very correct order. So, whenever you are dealing with such things, you should be extra cautious. Okay. So, microbiological waste. What are those things? Because whenever you are dealing with these things, what you see specimens, cultures, okay, discarded viruses. See, whenever these things, these experimentation procedures, and everything is going on, what you see and what you observe is that they are always dealing with the pathogens. When dealing with the pathogens and when dealing with such scenario at every level, one should every time be readily very careful in dealing with such scenarios, because if you don't deal with them, then they can turn out to be really very dangerous in a situation. So, pathological waste, unfixed human tissues, or waste biopsy material, like the things that are dealing with in OT, the operation theaters. Okay, any accident in cases, the waste they have to discard from the operation theater. Sharp waste, tubing needles, scalpels, all those things they come under these things of biohazardous material. So, waste bag classification. That every time you must understand that the waste bag classification depends on the color code. Okay, what type of color is being used in the scenario? Okay, what type of the things that are under process, and how you should understand the scenario that follows the color code. The testing, analysis, and classification of biohazardous waste are always critical. The waste bag classification system, which is issued by the government, gives guidelines on how waste should be sorted. So, what is the correct meaning of sorting? The correct meaning of sorting, that you have to separate out each and everything according to its predetermined or predefined scenario and protocols. Okay, it is not like that you can discard any waste anywhere. Every time you have to follow it, and every time you have to observe it, right? Gives guidelines on how waste should be sorted and temporarily stored before treatment and disposal. Okay, how you have to treat such a dangerous material, and how you have to uh, sort it out, and how you have to dispose it out. So there are protocols and color codes that should be followed every time. Orange bins are for the storage and disposal of sharps not containing or contaminated with medicines. So in orange bins, you have to discard the sharps that are not contaminated with medicines. For example, sharps for blood samples and acupuncture. So any sharp object that has been in contact with the blood sample or any acupuncture thing, then it should be discarded in orange bins. So here are the details that are essentially required when you are observing the biomedical waste management. Okay, so there are different types of colors that are readily available, and they represent different scenarios and different situations. Yellow bags. Okay, what they represent: infectious waste, bandages, gauze, cotton, or any other uh, objects in contact with body fluids, human body parts, placenta, etc. So here they are. What they are saying, they are saying that you always have to see the gauze where the things are placed properly. Infectious waste, okay, that are as a result of dressing and other things. Object that contains bodily fluids of human body parts, they all should be discarded in yellow bags. So when it comes to red bags, what are the things that are present inside it? Plastic waste such as catheters, the pipes that are used, injection syringes, tubings of and IV bottles. So every time such type of waste is present in the red bags or red color bags, blue bags, all types of glass bottles and broken glass articles. Okay, all the things that are linked with glass, bottles and broken glass articles, outdated and discarded medicines. So expired medicines, they all should be placed in blue bags. And black cardboard. What are the things? Needles without syringes, blades, sharps, and all metal articles. They all should be placed in black cardboard. So they can move further on, and they can readily be sterilized and further processed on for the recycling procedure. Yellow bins are used for the storage and disposal of sharp waste contaminated with or containing medicines or anesthetics. Red bags are for the disposal of solid or liquid items contaminated with blood or other waste with infectious material. Purple bins are for the disposal of sharps and medicines with cytotoxic or cytostatic content or contamination. So, purple veins are mostly seen or met in the microbiology labs, in the genome analysis lab, or all the labs that are working seriously on the molecular concepts. So here, there you can see many objects that are having cytotoxic nature, cytostatic nature. They are all discarded in purple bins. Okay, blue bins are used for the disposal of outdated drugs used drugs, denaturing kits, and discarded items used for pharmaceutical handling, including bottles or boxes, with residues, gloves, masks, connecting tubes, syringe bottles, drug vials, and anatomical waste. Every time all these things are discarded according to their scenario, like what type of waste should be discarded in what matter, and every time one must observe that these waste they are generated when they are generated since that very early beginning they are disposed of in their particular bags with having that are having a different color code, and they should always be kept in a separate container according to their hierarchy, and what type of waste should be dealt in what matter, that we will observe now. So, biomedical waste management. Here you can see the segregation of hospital biomedical waste. How one should deal with the hospital biomedical waste. So here you can see the first thing first is a green color. You can see general waste, kitchen waste, paper, and tissue and water bottles and cans. All such things they are disposed of in green color container, and when they are secured in landfilling, before that they are incinerated. Okay, so that the waste does not contain any pathological or infectious agent, right? So here in a similar manner, you can see the red color container, infected plastics, syringes, gloves, and plastic waste. Okay, so they are all stored in this type of container, and autoclaving is very necessary before securing them in uh, a landfill, right? So further on, you can see the yellow color bar, and in that thing, there what you can see that infected waste, soiled, anatomical, chemical liquid, cytotoxic, laboratory waste, expired and discarded medicines. Okay, so before that, they are also subjected to plasma pyrolysis, uh, in which the incineration is done in the absence of oxygen, and autoclaving is also preferred sometimes, and deep burial is preferred for such type of waste in a landfill. Glassware, antibiotic vials, metallic implants, glassware, material except cytotoxic. They are all placed in a blue color container, and they are passed on to the recycler for the recycling procedures. And autoclaving and incineration is preferred before being recycled, right? And sharps, when the container is of white color, it must be having sharps, needles, cuts, glasses. Okay, so sharp pit, they are disposed of. Sometimes metal objects are what you say subjected to the furnaces where they are melted down, and uh, other recycling procedures are commonly carried out. So common treatment facilities operate in such manner, and here they segregate the waste material, especially the biomedical waste material that is hazardous in nature, is discarded according to their hierarchy and according to the conscience of the protocols. So, waste auditing is also an important means of managing waste disposal and volumes and frequency of waste types. So, waste auditing is also essentially very important because whatever the type of waste is being discarded of or being uh, is being sent off should be monitored properly, and proper records should be maintained, and auditing should be done from time to time in order to see that everything is being operated in a very similar scenario. No procrastination or carelessness is being carried out, right? So, precise regulations for disposing of biohazardous waste are strictly controlled. Every time they are strictly controlled, and the rules are to be followed properly. No carelessness or any other such thing is entertained. Waste management is important for biohazardous waste to prevent diseases and infection risk to anyone who may come in contact with it. So, why this waste should be discarded off properly? Because this waste is biohazardous in nature. Okay, it is not only hazardous in nature but biohazardous in nature, and it can readily infect any individual who comes in its contact. So, it is not like that only okay, a healthy individual will not get infected. He or she may get readily infected because these things are uh, disposed of from very uh, what you say, from testing facilities, from laboratories, from hospitals and clinics. And there they are dealing with a lot of pathogenic diseases all the time, or the chemical experimentation that are linked with chemicals and biology or sometimes both. So these wastes are very dangerous in nature, and proper maintaining and auditing facilities should be maintained. Why is it so important to follow incinerator legislation? Okay, so why incineration procedures are very important? Though widely practiced, the incineration of waste has been inadequate or or the incineration of unsuitable material results in pollutants being released into the air and ash residue. So, most probably the best method is incineration. But the problem with incineration is that in long term, these things they do produce a lot of pollutants. When they are left in the air, they pollute the atmosphere also. So, whenever dealing with such scenarios, one must understand the proper protocols and every aspect of the things that are being explained and elaborated in a very simple manner. So one must understand how we should readily deal with such products or such waste material that is biohazardous in nature. Okay, so the incineration of heavy metals or material with a high metal content, in particularly lead, mercury, and cadmium. So, if the waste is having mercury portions, lead and cadmium, they are very much poisonous for the uh, living organisms, and they should be dealt in accordance. Can lead to spread of toxic metals in the environment, and they are really very toxic. And mercury poisoning or lead poisoning is very common. So, whenever you are having such chemicals, you should discard them properly and you should always refrain from discarding such materials in the environment. So, modern incinerators operate at operating at 850 to 1100 degrees centigrade. This is a very high temperature when they operate. They are almost like a furnace. Okay, fitted with glass cleaning equipment uh, are the only ones able to comply with the international emission standards of dioxins and furans. And most probably, when the incinerators are working, they should work properly. It should not be like that when you are trying to incinerate the product, the incinerator not working properly as they are breaking down. And you should understand at what temperature that particular thing or that very particular product should be incinerated. Okay, and what will be the byproducts and how that very particular product would be discarded in the environment so that it cannot be biohazardous in nature. Okay, informing your local authority and environmental agencies of what material you are burning is vital. Once aware, they can approve or disapprove their incineration and to avoid any environmental concern. It is important to follow regulations for biohazard waste disposal every time. So, what are the benefits of incineration? Due to high temperatures during incineration, chemicals or any contaminated supplies that pose a risk to health can be destroyed. Okay, most of the time the chemicals they also get denatured very easily, and they do uh, cause them a lot of damage to the integrity of the chemicals in very high temperature, and they get destroyed. Okay, but any contaminated supplies that pose a risk to human health can be destroyed also because high temperature it just denatures everything. Incineration ensures the complete destruction of hazardous waste that would otherwise go to a landfill, which can be very dangerous as there is a risk of ground pollution. So, in long term, somewhat incineration is practiced and preferred really safe because the procedure of landfilling is really very dangerous when it is considered because in the long term it does produce a lot of problems and a lot of scenarios that do reveal that this practice is much more safer than compared to the landfill because Landfills, the waste material can seep out and seep inside the Earth's surface and reach the water table also. That can easily lead to ground pollution. Infected waste can pollute the ground, which could enter the food and drink supply. This is the main thing that I was trying to explain it to you. Why choose incinerator? Knowing the importance of promptly providing the right products to the location, Incinerator 8 have many years of biohazardous emergency. And when you have very good incinerator, they do provide you easy solution for discarding of this biohazardous waste material that is extracted from hospitals, that is extracted from research laboratories simultaneously. Waste source, waste at a source, waste transport, waste treatment disposal, and all those things now we are going to explain. So, waste source on that very particular site, assessment is done, waste reduction program is carried out. Okay, how you should adopt the practices like where you can reduce the amount of garbage being produced, right? Uh, and especially the biohazardous garbage, identify recycling opportunities, and reuse the waste. So these are the safe practices that are very much appreciated in the long term. Waste transport, proper labeling, contamination proofing, regular testing and monitoring. Whenever the waste is being transported, proper auditing is required, and auditing is done in order to check the labeling has been done properly, contamination proofing has been done, regular testing and monitoring is always required because you always have to check the individual is working properly or not. Waste treatment disposal, where mostly you can see the recycling procedures also included in that. Reduce waste to landfill, waste to energy, waste incineration, waste solidification. So these are some of the methods that are essentially adopted in order to reduce the biohazardous waste because most of the time the waste is being reduced to a landfill. Waste is converted to a source of energy for power generation. Waste is reduced by incineration and also by solidification, and its solids are being used for the construction of roads. Also. Biohazardous waste categories. There are four general categories of biohazardous waste based on physical form of waste. Each form must be segregated, identified, decontaminated, and disposed of in an appropriate manner for the form in order to minimize occupational exposure and environmental risk. So, this protocol is essentially very important and it must be followed in order to avoid the environmental accidents. So every time the sorting is necessary, segregation is very necessary for the waste material identification. This is this type of waste material, and this should be treated like that. Decontamination, okay, mostly pyrolysis, incineration, and autoclaving is carried out and disposed of in an appropriate manner so that it cannot be great environmental risk or any sort of it can pose any sort of risk to any individual also. Biohazardous waste in any form should not be left unsecured in areas that are accessible to the public. That is left in hallways and open grounds or any other things. So, whenever you are dealing with biohazardous waste, it should never be left unsecured outside in the open or in any other hallway, so that some accident can also take place, but other individuals can also come in contact with that very particular waste. Only lab personnel should remove biohazardous waste from the lab area and transport it to waste holding areas for final disposal. So laboratory people are only in charge of handling those sensitive biohazardous laboratory waste, and it should be done properly and in order to avoid any other sources of cross contamination or outbreak. So solid biohazardous waste that mainly contain non-sharps, soil collection, treatment and disposal, liquid biohazard waste, and sharp bottles. So when you see what is a hazardous waste, there are many types of classification and they have changed over time. But you can say that anything that is uh, poisonous in nature is a hazardous waste. So disposal of for non-sharp and sharp biohazardous waste. What is the basic procedure? Collect bio-waste at the laboratory level in designated biohazardous label cans per current bio-waste collection procedures. Segregate serological packets and pipette tips to prevent bag puncture or tears. Collect biohazardous sharps in sharp containers as required. The following are acceptable waste: stock, propagated culture of infectious agents, materials that have been used for the collection, processing, storage of human or animal blood or bodily fluids, including cell lines, recombinant synthetic nucleic acids, or lab consumables, contaminated with any of these materials. Following are unacceptable waste. Hazardous chemicals, for example, phenol, chloroform, aerosol with ethylene bromide, radioactive waste, bulk liquid waste, pathological waste such as human, animal bulk, blood, tissues, or animal carcasses. Okay, for biosafety guidance, human fetal remains, limbs, or cadavers, that means dead bodies, compressed gas cylinders, loose shafts, controlled substances, etc. So once the bag is full, two-thirds full, close the bag by gathering the top, twisting and closing with a single overhand knot. This method of closure minimizes the risk of leak, spills, and is required by uh, DOT since material will be transported in commerce. Do not tie the bag closed by crossing tabs. Okay, by buying your dog here method. Okay, that is not at all applicable and practical. So here you can see in this image how to close the bag properly. How you have to twist and put a knot over it, right? Overhand knot. So gather a top bag and twist it. Make a loop, then push the bag and pull on the bag to tighten the knot. So do not close the tab in the bunny or dog ear method. Yeah, it should not be practiced like that on the extreme right, so that because of this they can frequently cause any sort of leakage or contamination. So moving ahead, once the bag is properly closed, double bag uh, with using the same closure technique listed above. So every time you have to make sure that bag is being properly closed, bag is being properly used, and bag is being properly operated following the protocol. The bag then is disposed deposited directly into the medical waste contractor provided acceptable 90-gallon bin uh, that has been emblazoned with company's name, the Universal Biohazardous symbol, that is UN shipping identifier UN 3291 and proper shipping name, okay, regulated medical waste NOS. So every time the protocols are followed internationally and nationally, they are very much identifiable. They are very much understood, and everything is followed should be followed according to the scenario. Because in the long term, these scenarios they do play a proper role in the safety and biosafety from this hazardous material that is readily available, and if not handled properly, it can also cause some type of cross contamination and outbreak also. Again, autoclaving bags prior to disposal will not be required except for following substances. And what are those substances when autoclaving is necessary? Any infectious agent listed on for UV, this means that stored or propagated cultures of pathogenic microorganisms like E. coli, Shigella, and hepatitis virus are to be autoclaved for minimum 30 minutes. So for the time duration of 30 minutes, they should always be autoclaved properly. Once treated, allow the bags to cool to room temperature, placed in a second bag, secured according to the above technique, and then discard into a medical waste contractor provided receptacle. So every time you have to follow the protocols in order to maintain the safety and integrity of the whole procedure. Any material under federal permit requiring on-site inactivation is an approved and validated material. There must be autoclave according to the validated parameters. Once treated, allow the bags to cool to room temperature and then discarded into medical waste contractor provided receptacle and dispose disposed of in a dumpster. So after following that procedure, you can easily dump off the things and easily get rid of the waste. So if you have not done properly according to the scenario, if any auditor or uh, medical practitioner is caught not following the protocol, he or she would be punished severely for putting others' health at risk and being careless. Properly packaged and permanently closed sharps containers may also be discarded into the medical waste uh, contracting receptacles. So everything should be packed up properly, everything should be maintained properly in order to maintain the integrity of the whole protocol. Sharps container must be secondarily enclosed in a secure type as described above biohazard bag prior to disposal. So every time they have to look into the matter and everything should be disposed of properly, and prior to that it should also be securely stored in a bag for disposal um, purposes. Permanently closed sharp containers must be wiped down with disinfectant prior to removal from the lab for the disposal. If there are any liquids present in biohazardous sharps container, it must be placed in a leak-proof secondary container and with a secure lid and a biohazardous label identifying the things inside it for transport to the to the waste collection site or prior to bagging from depositing in a medical waste contractor bins. On-site autoclave treatment if required. So whenever you are dealing with these type of waste material, it is very much necessary for you to understand the scenario. Autoclavable waste bags must be used in biohazardous waste collection containers. Bags must be placed in a secondary container, that is, a tray with raised slides, which is placed on a cart for movement to the autoclave facility. Practice notes on biohazard bag. Biohazard bags are a one-way means of disposal. Do not dump the contents uh, from one biohazard bag into another, as this action spreads contamination and increases your exposure to this waste. So every time you have to look into the matter for not only your safety but also for the safety of others. Biohazard bags need to be contained at all times during the collection, treatment, and disposal process. Some lab items may puncture bags, and this can lead to leaks and spills. Bags awaiting autoclave treatment should be stored in trays, tubs, and buckets. The only exception to this practice is when small quantities of biohazardous waste is present there that do not contain liquids are collected temporarily in bench top containers. Biohazard bags must not be used for the collection of other hazardous waste, that is ethylene bromide gels. So every time you must be properly educated and informed that how you should carry out the scenario, and you should understand the gravity of the situation every time. So these are the MCQs that you have to practice now. Some of the difference between a fume hood and biological safety cabinet (BSC) and the are that a BSC from protects both the user and the material inside the cabinet, and that the exhaust is HEPA filter. That is two of things. PPE is personal protective equipment, public protective equipment, possible protective equipment, or all the above. Which of the following items is considered sharp? Needles, scalpels, microscopic slides, and cover slips, or all the above? Laboratory working with human cell lines must display the biohazard hazard symbol on the areas where the work is performed and materials are stored. That is true or false. UV light can be utilized as the sole form of decontamination in biological safety cabinet. It is true or false. Hands should be washed before and after working in a biological safety cabinet. True or false. Which of the following practices should be utilized when working in biological safety cabinet? Okay. Disinfect the work surface of BSC with uh, before and after work. Disinfect all items which go into and come out of BSC. Do not store any item in BSC. Allow the BSC to operate before work begins and after work classes, or all the above. Which of the following practices are not allowed in the laboratory? Eating and drinking, applying cosmetics, handling contact lenses, or all the above. It is appropriate to wear two gloves and carry loose tubes in the elevator. True or false? When it is hot outside, it is okay to wear shorts and sandals to work in the laboratory through our phones. So these are the references from where we have collected the information. And thank you very much for listening us.